Hey, welcome back. So we are going to talk a little more specifically about G protein coupled receptors. These are a really important category of receptors that as you continue your biology education, you'll probably explore in more depth in future classes in grad school. So we're just going to briefly introduce them here. The G protein coupled receptors, or often they're abbreviated as GPCRs, are named that because when the receptor is activated, it changes a G protein. So the receptor is coupled to a G protein. That G protein is going to be something that binds to a guanine nucleotide. So it's the receptor coupled to a G protein, which is going to have either GDP or GTP bound to it. One in particular that has been studied a lot is the beta-adrenergic receptor. As you look at this, you can see that these receptors have the C terminus in the cytosol. Then they have 7 transmembrane sequences. So 1, 2, 3, 4, 5, 6, 7. And then out, that N terminus is extracellular. And although they all have pretty similar structures like this, they have pretty different actual amino acid sequences. So they'll be able to bind to different ligands on the extracellular side and different effector proteins on the cytosolic side. So when an activated G protein binds to its target protein, it'll alter its activity. These will include things like your olfactory receptors, the beta-adrenergic receptor, and hormone receptors. Another really important category of receptors here would be the opioid receptors. These are part of the G protein coupled receptors. Opioid receptors are what narcotic drugs like morphine bind to. So G protein coupled receptors all have a pretty similar structure, different amino acid sequences. They form 7 transmembrane alpha helices with extracellular or intracellular loops on them. The extracellular portion has a unique messenger binding site, a unique ligand binding site, and the cytosolic portion will allow it to interact with only certain types of G proteins. Those G protein coupled receptors can be regulated in a few different ways. One is through phosphorylating their cytosolic terminus, their C terminus. So GRKs are G protein coupled receptor kinases that can, when there's an active G protein, they phosphorylate it. And then when it's phosphorylated, that binds to a molecule called beta-arrestin. Beta-arrestin, when it binds, will inhibit that G protein. Okay, so your G protein bound to a ligand, it's active, and now it's time to be deregulated. The GRK phosphorylated it. The phosphorylation allows the beta-arrestin to bind, and the whole signaling message is going to be inhibited. So we're able to modulate and control our G protein coupled receptors through this pathway. G protein kinase A will be activated by G protein mediated signaling. So when the G protein is active, protein kinase A will be active, and protein kinase A, being a kinase, will phosphorylate. It can phosphorylate other amino acids on the receptor and also inhibit it. So this is a type of negative feedback. When the receptor is increasingly sending a message, the protein kinase A or the beta-arrestin are going to be increasingly down-regulating it. The signal and the effect are going in opposite directions. Okay, but when we are not turning it off, when we're using our G protein couples, our G proteins are going to be molecular switches between an on and an off. When they are bound to GDP, they are off. When they're bound to GTP, they are on. G proteins can either be large heterotrimeric G proteins or small monomeric G proteins. Your book really focuses on the large heterotrimeric G proteins, so that's really what we're going to talk about. They're called heterotrimeric because they're made of 3 different subunits. They have a G alpha, a G beta, and a G gamma subunit. And the association of those 3 subunits is what will be affected by phosphorylation of GDP, and that's what will then signal for other things to happen in your cell. So G alpha is the largest subunit of the G alpha beta gamma trimer, and it binds to a guanine nucleotide, either GDP or GTP. When the G alpha is bound to GTP, when the ligand is bound, it causes the alpha subunit to bind to GTP. It's going to detach. So your alpha bound to GTP has left the beta gamma subunit, which stay tightly, permanently stuck to each other. And this can either then stimulate a signal transduction or inhibit a signal transduction pathway. If this is going to turn on a pathway, it's going to be called GS for G stimulating. And if it inhibits it, it's going to be called GI for inhibiting. So this can be an inhibitory or stimulating pathway. When the messenger binds to the GPCR on the surface, the receptor changes conformation and causes the beta and gamma subunits to associate with the receptor, and the alpha releases GDP and binds to GTP. G alpha will pick up a new GTP molecule and detach from this beta gamma complex. Depending on what kind of G protein it is, what type of GTP alpha or beta gamma we have, it can actually initiate that signal transduction event by itself. Then each portion of this G protein, the alpha or the beta gamma, can then exert an effect. So it'll bind to another enzyme, it'll bind to another protein, and this signal is going to continue as long as G alpha is bound to GTP and G alpha and G beta gamma are separate. The G alpha subunit does catalyze GTP hydrolysis, so it's only going to be active as long as it's bound to GTP. Once that GTP has been hydrolyzed, it's back to being GDP. That alpha beta gamma subunit all reassociate with each other. That shuts down the signal transduction pathway, and when the messenger is no longer being sent. G protein activity is going to be enhanced by regulators of G protein signaling pathways, or RGSs. These are GTPase activating proteins that are important in activating and regulating G protein function. And the most important G protein function is to release second messengers or form second messengers. So far you've had a receptor bind to a G protein coupled receptor. A ligand bound to a G protein coupled receptor. That then triggered your heterotrimeric alpha to switch out its GDP for GTP. So you have an alpha and a beta gamma, and they're going to do something starting with a second messenger. So the alpha subunit of activated G proteins can activate with and interact with other proteins. Some of the things it can interact with would be adenylyl cyclase. There's an important one we're going to talk a lot about. And phospholipase C. There's an important one I think we've already talked about a lot. And G protein receptor kinases can also be activated by the beta gamma subunits. Here, the beta gamma subunit is able to open this ion channel. So one example of signaling with the beta gamma subunits would be the muscarinic acetylcholine receptors. So when acetylcholine binds to the muscarinic acetylcholine receptor, the beta gamma subunit is going to dissociate from the alpha as the alpha is bound to GTP, and that dissociated beta gamma subunit are going to associate with the ion channel and cause it to open. When acetylcholine is no longer present, it'll trigger for alpha to release GTP, hydrolyze it back to GDP, and alpha beta gamma will all interact again. So cyclic AMP is a pretty important second messenger. There's a lot of different G protein coupled receptors. So there's a lot of different G protein mediated signal transduction events, but the most important ones and the most common ones are going to involve second messengers. So that means receptor ligand binding, G protein activated, and then something else, a second messenger there. The most common second messengers are cyclic AMP, which is what we've got going on in this image, and calcium ions, both of which are important. Both cyclic AMP and calcium ions are important second messengers, and we're going to come back and visit both of them by the end of this unit. Cyclic AMP is a second messenger that is going to be formed by this enzyme adenylyl cyclase. So you have ATP. It can dephosphorylate, hydrolyzes ATP down to AMP, and then trigger it to cyclase. So this phosphate is causing this cycle to happen. That's what adenylyl cyclase does. This enzyme, adenylyl cyclase, is this blue little Pac-Man guy here that is going to be active until that stimulating alpha portion of the G protein binds to adenylyl cyclase. Then it will activate your cyclic AMP. And now you have your second messenger ready to go send the signal. Okay, I'm going to take you through a walkthrough of this figure, and then I will come back and talk to you in our next video talking about what happens once you have your second messenger like cyclic AMP activated. Okay, give me a second to come back. Okay, so this is within the mastering program, and I've told you before how much I love the mastering products. So hopefully you have gotten a subscription to this. It is a great study help. But let's walk through this figure.
Hi, this is Jeff Harden from the University of Wisconsin in Madison, Wisconsin. Today I'm going to walk you through this figure, showing one way that extracellular signals can be transmitted to the interior of a cell through a typical G protein coupled receptor. In this example, we'll see how G proteins can cause the formation of the important second messenger, cyclic AMP. To orient you, part of the plasma membrane is visible here, defining the inside and outside of the cell. You can also see a number of proteins are embedded in this membrane. The first of these is the enzyme adenylyl cyclase, which is responsible for the formation of cyclic AMP from cytosolic ATP. Normally, it is inactive, with its catalytic portion protruding into the cytosol. Next, let's examine the multi-subunit G protein. The G in G protein stands for guanine nucleotide binding protein. G proteins act very much like molecular switches, whose on or off state depends on whether the G protein is bound to GTP or GDP. Since this particular G protein is bound to GDP, it is in the inactive state. Pictured in this figure is one class of G protein called a large heterotrimeric G protein because it contains three different subunits, G alpha, G beta, and G gamma. G proteins that inhibit signal transduction are called GI proteins, and those that stimulate it, like the one illustrated here, are called GS proteins. Therefore, you are looking at an inactive, large, heterotrimeric GS protein. Finally, let's examine the receptor that will initiate the signal transduction pathway. Since this receptor interacts with the G protein, it is called a G protein coupled receptor, or GPCR for short. You're looking at a simplified view of this receptor. Let's see what it looks like in more detail. The receptor forms 7 transmembrane alpha helices connected by alternating cytosolic or extracellular loops. The N-terminus of the protein is exposed to the outside of the cell, whereas the C-terminus is inside. The extracellular portion of each GPCR has a unique ligand binding site, and the cytosolic loops allow the receptor to interact with the G proteins. Zooming back out, we see where the ligand binding site is on the GPCR. This is where the ligand molecule will bind and initiate signal transduction. When a receptor is activated by ligand binding, the receptor-ligand complex associates with the GS protein, causing the displacement of GDP by GTP and the dissociation of the GS-alpha GTP complex. The GTP-GS alpha complex, now in its active form, binds to membrane-bound adenylyl cyclase. This activates the catalytic domain of adenylyl cyclase, leading to the formation of cyclic AMP from ATP. Activation ends when the ligand leaves the receptor. The GTP is hydrolyzed to GDP by the GTPase activity of the GS alpha subunit, and the GS alpha subunit dissociates from adenylyl cyclase. Adenylyl cyclase then reverts to the inactive form. The GS alpha subunit eventually reassociates with the GS beta gamma complex, and cyclic AMP molecules in the cytosol are hydrolyzed to AMP by the phosphodiesterase enzyme. This shuts down the pathway promptly when the concentration of the ligand outside the cell declines. An increase in cyclic AMP concentration can produce different effects in different cells. In skeletal muscle and liver cells, the breakdown of glycogen is stimulated. In heart muscle cells, the contraction gets stronger, whereas in smooth muscle, contraction is inhibited. Cyclic AMP blocks mobilization of platelets during blood clotting, and in intestinal epithelial cells, it causes secretion of salts and water into the lumen of the gut. As you can imagine, defects in this pathway can have a significant impact on human health and disease. Thanks for watching, and good luck with your course.
Okay, so I'll see you back in a little bit to carry on what happens in our messaging here. See you later. Bye.